D-aspartate is one of the most abundant free D-amino acids in mammalian tissues, and in the central nervous system it is involved in glutamatergic transmission since it can activate several receptors. The dysfunction of glutamatergic transmission is linked to the onset of several neurological and psychiatric disorders. An alteration of D-aspartate levels has been reported both in the brain of schizophrenia patients and in an animal model of autism spectrum disorder. Since D-aspartate synthetic pathway is still unclear, D-aspartate oxidase is the only enzyme known to control the endogenous levels of this molecule. The aim of this project is to evaluate the processes involved in the regulation of D-aspartate metabolism under both physiological and pathological conditions. 5 research teams with different and complementary expertises will collaborate to accomplish several tasks. Inhibitors of D-aspartate oxidase will be identified by virtual screening at the University of Milan. This in silico approach will be used to screen several virtual chemical libraries of low molecular weight compounds. The best identified inhibitors will be then synthesized and chemically optimized for further structural characterization, and finally validated on cellular lines ectopically expressing this protein in collaboration with the University of Insubria. Both teams will collaborate to identify the proteins that interact with D-aspartate oxidase at the cellular level. At the University of Tor Vergata, we will perform an in-depth analysis of the effects of DDO gene dosage on synaptic transmission and neuroplasticity in ex vivo brain slices preparation. Specifically, we will use electrophysiological techniques to measure long-term potentiation in the hippocampus, as well as excitatory glutamatergic and inhibitory GABAergic transmission and the excitation-inhibition ratio in the prefrontal cortex from DDO knockout and DDO knock-in mice at different age stages. On the same models, we will also investigate the structural synaptic plasticity features by analyzing dendritic synaptic morphology at different levels of complexity. Finally, the electrophysiological profile and the pharmacodynamic properties of novel DASPO inhibitors on ex vivo hippocampal slices will be investigated. At the University of Campania, we will evaluate whether DASPO modulation, obtained either through a genetic approach or based on the oral administration of the newly identified DASPO inhibitors, affect behaviors relevant to autism by assessing social skills of mutant mice through the analysis of the social interaction test, in which the duration of social interactions and aggressive behavior are analyzed as an index of spontaneous social interaction skills. We will also evaluate sociability, social novelty preference and social recognition memory skills of mice by performing the 3-chamber test. The mice brain morphology and function will be evaluated at CNR by in vivo imaging. With magnetic resonance imaging we will study the anatomy of the brain and compare the volumes of gray matter, white matter, and cerebrospinal fluid between different groups, performing also voxel-based analysis to highlight regional significant variations of brain volume. We will also study the brain functionality with resting state functional magnetic resonance imaging. Finally, the University of Insubria will perform a chiral amino acid analysis of blood samples of patients with an autism spectrum disorder diagnosis. The outcome of this project might allow to clarify the mechanisms that control D-aspartate metabolism and to shed light on its role in modulating the glutamatergic processes that regulate brain development and function, even during the onset of disorders such as schizophrenia and autism.